कम्प्युटर ऑडिओ नाही करत आहेस. होस्ट मी. होस्ट मी
आहे ऑलरेडी इथे. येतोय.
थांब मग मी करते, व्हॉल्यूम करते 1 मिनिट. हॅलो,
हां.
आता ओके आहे?
तरी थोडंसं व्हॉट अबाउट
अजून करू?
हो हो.
हा हा, व्हॉल्यूम नाही येते तिला.
तो माइक
हाँ
म्हणजे पकडता का?
मैम हम लोग को क्लियर आ रहा है ऑडियो.
हाँ पंकज,
मैम हम लोग को क्लियर ऑडियो आ रहा है मैम का.
आ रहा है?
ठीक है. तो हाँ, मेरा आ रहा है ना क्लियर?
यस
वॉल्यूम
मैम.
आ रहा है लेकिन.
Okay. Good morning, you all. Say all
Morning,
of you
ma'am.
good morning. Good morning. Everybody's there. Hello.
मॅम थोडंसं
काय करू थोडं?
इन्क्रीज करा व्हॉल्यूम, कारण आहे ना लॅपटॉप मध्ये हे रेकॉर्डिंग व्हायला नाही तर त्रास होईल.
हा, काय करू, वाढवू व्हॉल्यूम?
हा, माईक जवळ पकडा.
बरं. ओके.
माझ्या मोबाईलवर व्यवस्थित आहे.
ओके.
इकडे नाहीये.
Okay. Yeah. Okay. Thank you. Good morning, all of you. I hope you are safe and fine. Now let us begin with today's lecture. Today's lecture, I will be doing a bit revision of pyrimidine biosynthesis, what we had done last time, and the new part also we will be discussing. So today we are doing pyrimidine biosynthesis. Now you all know very well that DNA is nothing but the heterogeneous polymer of nucleotides. These nucleotides are composed of the 3 units, of which nitrogen bases are most important. The other components are sugars and phosphate molecule. Now, when you look at the nitrogen bases, you know very well that these nitrogen bases are of two types, that is purines and pyrimidines. Purines, the entire account we will be seeing in the next few lectures, but today we are discussing pyrimidine and its biosynthesis overall. Now, students, if you look at the screen, what we had done last time, what we had done is, we had done the pyrimidine biosynthesis, and there we have learned few things which were, just 1 minute, my things, just 1 minute. Just 1 minute, there is some, it is not good. Okay. So here we had seen the pyrimidines. Now students, what I want you all is, remember these 3 structures very well, because I'm going to give you the reference of this structural aspect repetitively. Okay, so there are 3 pyrimidines, what we call as the cytosine, thiamine and uracil. Now cytosine is common for DNA and RNA. Now students, pay attention to the chemical name, the chemical nomenclature. Cytosine is 2-oxy-4-aminopyrimidine. Cytosine, if you see here, you can make out that the oxygen is at the second position, as well as, at the second position, and at the 4th position, there is an amino group. So here you will find the cytosine is named as 2-oxy-4-aminopyrimidine. Now, if you come to the next, and that is thiamine. Thiamine is 2 and 4. That is the second position, as well as on 4th position, there is an oxygen. And at the next position, and that is 5th position, there is a methyl group. So what we call it as 2,4-dioxy-5-methylpyrimidine. Now students, you know very well that this is the component of only DNA. This is not present in RNA molecule. So this is the unique one. We will be learning biosynthesis of thiamidine separately. Now here the chemical name I'll repeat, and that is 2,4-dioxy-5-methylpyrimidine. And the third one, which is the absolute component of RNA, and that is uracil. Uracil is named as 2,4-dioxypyrimidine. So oxygen is at second position as well as at the fourth position. I will remind you once again that pyrimidines are the nomenclature is done clockwise, considering the atoms starting from nitrogen. So this is first, then this is second, then this is third, fourth, fifth, and sixth. So this type of nomenclature you are going to remember very well. Now, one more important point I want to tell you is when we will learn biosynthesis, the most important intermediate which is formed is the nucleotide with uracil, what we call it as the uridine. You have heard this. Now this uracil, when it undergoes amination, it will form cytosine. Whereas the uracil, when it goes methylation, it will form thiamine. So remember that very well. So these three are comparatively easier to remember, and remember one thing, that uracil is the major intermediate in the biosynthesis pathway, and that later on gets converted as per the requirement to cytosine, and then with few more changes to thiamidine. So these are the three pyrimidines which we get in our DNA and RNA as an important component. Students, just I'll give you few seconds to please focus on the structure, because otherwise you will not be easily understanding the biosynthesis pathway at all. So there are three pyrimidines, I said: cytosine, thiamine, and uracil. Now these are the nitrogen bases. Basically, these are the nitrogen bases. These nitrogen bases, when they are attached to the respective sugars, when these nitrogen bases are attached to the respective sugars, that is ribose sugar or deoxyribose sugar, at the first carbon position, it forms nucleoside. What it forms? It forms a nucleoside. And when this nucleoside is connected with the phosphate group, then we call it as a nucleotide. So as I had mentioned last time also, that the phosphate and sugars in a particular molecule, that is DNA or RNA, are common. What differ is the nitrogen base, whether it is purine or pyrimidine. So these are the three pyrimidine structures. Now look at this. Now look at this structure. What this picture tells you. See here, this is the basic pyrimidine ring. This is the basic pyrimidine ring which has been shown. As per the requirement, when a particular pyrimidine is synthesized, the required atoms are added. So whether it is amino group, whether it is methyl group, or whether it is oxygen, according to the requirement of formation of particular pyrimidine, these atoms will be added during the biosynthesis. But this is the basic ring of pyrimidine. Now pyrimidine is a single ring structure. They are the smaller molecules, and they contain nitrogen only at first and third position. Remember this again very well. So pyrimidines are single ring structure. They are the smaller molecules, and they contain nitrogen at first and third position. Now here, what I have tried to show you is what are the general sources. So here you can see at the second position and at the third position, these carbon and nitrogen and other required structures are donated or obtained from the carbamoyl phosphate molecule, which is formed from the glutamate and carbonic acid. That also I will show you in the next slide. And the remaining part, see here, the position that is at first, fourth, fifth, first, fourth, fifth, and sixth. This is obtained from aspartic acid. So aspartic acid contributes maximum to form pyrimidine ring. Now here, see, you have to remember this line also very well. Pyrimidine is first synthesized. I'm talking about the biosynthetic pathway when you are studying that. So pyrimidine is first synthesized and later it is attached to ribose phosphate. So in pyrimidine biosynthesis, these are the key points which you are going to remember. And that is, in pyrimidine biosynthesis, pyrimidine molecule is first synthesized independently, separately, and then it is attached to the respective sugars. So whether it is ribose or deoxyribose, then it is attached to the sugar. Now the same part or same illustration, but probably this may be the simpler for you. And see here, this second position and third position. Second position, that is a carbon and hydrogen will be there. Then at third position, nitrogen. Nitrogen is obtained from glutamine. It is in that image, it was L, but it is T. So from glutamine, and then second position carbon is obtained from carbonic acid, whereas these all remaining ones, they are obtained from the aspartate itself. So this is very important. If you understand this very well, and if you remember this very well, biosynthetic pathway should be very, very easy for you. Very easy, believe me. Even the structural changes you will be able to remember very well. Now students, let us come to the revision, because I had mentioned that last time, but anyway, we will study now this once again. Now here I'm showing you the biosynthetic pathway, which is a de novo pathway. Now this de novo pathway, when you talk about de novo pathway, what you are going to remember is, okay, just take few seconds to look at the reactions from left side. So here, when I talk about the structures, you have to, okay, so let it be. Just have a look at the first one, okay. Now this is the de novo pathway I'm showing you. De novo pathway is the pathway in which the pyrimidine rings are biosynthesized from the individual atoms or individual substances. It is totally formed, it is totally constructed, and then it is attached to the sugar. So it is a long pathway and an expensive pathway for a cell too. So let us start first with the reaction. Now here I would be explaining you the details. Now here what I have done, whenever I've selected the image, what I've done is, see, the names which are written by red, I have indicated the part which is donated by that particular substance also in the same color. So I think that should be very easy for you. So the first step is, see, CO2 plus glutamine. Now carbon dioxide plus glutamine, and this carbon dioxide, when it enters into the cytosol, of course, it forms a carbonic acid, that is HCO3 minus. But here, just for the simplification, let us consider carbon dioxide plus glutamine. It comes in the picture, and this glutamine gets converted into glutamate. It donates its amino group. Here, 2 ATPs are used. So energy currency is used. So 2 ATP molecules are converted into 2 ADP. Phosphate is released. This phosphate is attached here, and now the compound is carbamoyl phosphate. Now this step is important for one more reason, students. This carbamoyl phosphate or this carbamoyl phosphate synthetase 2, what we call it as CPS2, this enzyme is a key enzyme. And when we will learn regulation, that time I will mention again the importance of this enzyme. So carbamoyl phosphate synthetase 2 or CPS2 is the key enzyme which shows the effect under its regulation. So here you can see the compound which is formed is carbamoyl phosphate. Now the next very important step 2. This is also a very important step where the aspartate comes in the picture. Now this aspartate, when it comes in the picture, the enzyme is aspartate transcarbamylase, ATCase. So ATCase or aspartate transcarbamylase is also a regulatory enzyme. Remember that. So step 2 is also a regulatory step, and this enzyme shows the control or activity depends upon the various factors due to which the entire metabolic pathway is under the regulatory mode. So here the aspartate comes in the picture, and see now this portion which is shown by the same color here, it is entirely donated. We had seen in earlier diagram that the position at the first, then this is the fourth, fifth and sixth, whichever. This entire portion is donated by aspartate. So here the entire portion, which is a major portion, is now donated by aspartic acid or aspartate, and now the compound form is carbamoyl aspartate. So this is the second compound which is formed in the pathway. Now comparatively the pathway goes very smooth and simpler. The third step which I'm showing here is, see, water is given away. The enzyme is dihydroorotase. The enzyme is dihydroorotase. Now here, students, please very carefully focus on this portion was already there. This particular region or this particular group is donated by aspartate. Water comes out. Now water is formed from this H, this one H, then this one H and this O. So this OH plus one H from here forms water, and that water goes out, and that is how it is, water molecule is exiting, leaving the cycle, and it forms now dihydroorotate. Did you notice one thing is, now here the aspartate part or the component which is donated is still a major one. So here what you get is, see, at the first position nitrogen, then carbon, then again nitrogen, C with double bond O, here also C with double bond O, so dioxy is already formed, and CH2, CH and COO. So it is still with the negative charge, and it is now dihydroorotate. Okay, so still it is on the way to complete the synthesis of pyrimidine ring. Now let us come to the next step. Now this step is, students, very important. Now here listen. What happens is the NAD gets reduced to NADH, and the enzyme is dihydroorotate dehydrogenase. So from where this hydrogen comes out? The hydrogen is from the CH2 is converted to CH. So one hydrogen has come out here, and this hydrogen is now placed to form a stable molecule, and that is COOH. We can call it as the orotic acid also. Now, when this hydrogen is coming out, here you will find it gets reduced and the free proton is also formed. But this free proton itself is incorporated in the substance. So I will show you again here. Just check. In this dihydroorotate, what you get is CH2CHCOO. Here what I get is CH, C and COOH forming the orotate or orotic acid. Now this is comparatively the stable molecule. So when we look at the next step, now come to the right hand side of this picture, of this diagram. Now this here, look at this. Now once the orotate is formed, last time I had mentioned, once again I will mention, this orotate formation is the key step towards the formation of pyrimidine ring. And now the PRPP comes in the picture. What is the PRPP? Phosphate ribosyl pyrophosphate. 5-phosphate ribosyl pyrophosphate. Pyrophosphate means these two phosphates are attached at the first carbon of ribose sugar. If you remember the structure, which I will be showing you once again later on, but this is phosphoribosyl pyrophosphate. So these two phosphates are attached to the first carbon atom of ribose sugar, which are to be displaced later on to allow the placement of pyrimidine ring. Now here the two phosphates are released and the enzyme are orotate phosphoribosyl transferase. So ribosyl pyrophosphate is now transferred, and here you will find, see now, our pyrimidine ring, almost formed pyrimidine ring is attached to the ribose 5-phosphate. So this ribose 5-phosphate is combined, or rather pyrimidine ring is attached to the ribose 5-phosphate with the help of orotate phosphoribosyl transferase. Now the next step, here we go for the next step. Now here if you see very carefully, because of the attachment of this ribosyl pyrophosphate and the release of this pyrophosphate, what you get is here orotidine monophosphate. So one phosphate with ribose sugar makes it monophosphate, and this is the complete ring of the orotate. When orotate is attached to the ribosome, obviously we call it as orotidine. You have heard uridine, cytidine, then thymidine. Similarly, it is an orotidine. It's a basic precursor of the required nucleotide. Now this orotidine monophosphate, very important step, decarboxylation. Decarboxylation takes place, and just have a look at this. The CO2 is gone. CO2 is released, and we get this ring as the uridine attached, that is uracil attached to ribose 5-phosphate. What was the name of uracil? You had said that is 2,4-dioxypyrimidine. So see now at the second position, at the fourth position, oxygen are present. The ring is completed and attached to ribose 5-phosphate or ribose monophosphate. So here what we get is the first product, uridine monophosphate. Now students, just take few seconds once again and see the steps of these two, this step as well as this step, till the formation of uridine monophosphate occurs. I'll just give you 5 seconds, have a look properly. Okay, so what has happened? From orotidine monophosphate, it is forming now uridine monophosphate by decarboxylation. By decarboxylation. So here, now UDP is formed, right? So UMP gets phosphorylated. That UMP gets phosphorylated. ATP gives away the phosphate. ADP is formed. Enzyme is simple kinase, and it forms UDP. Now this uridine diphosphate is a very, very important intermediate. Later on, we will be learning how this affects the regulatory steps also. So UDP is the intermediate now which is responsible to form the remaining two pyrimidines. Now students, very carefully pay attention to this bifurcated pathway. This pathway we are going to study separately. I will be showing you the illustrative slides picture, how this thymidine monophosphate, that is the pyrimidine required for DNA molecule, is formed. Okay, so that we will be learning in detail. And here you will be looking at this portion for understanding how the formation of cytosine or cytidine triphosphate ultimately is formed. Now here I would like to explain you few important steps. This uridine diphosphate undergoes once again phosphorylation, that is ATP is utilized, enzyme is kinase. Twice it occurs as a very important steps. So UDP forms the UTP. that is uridine triphosphate. Now this uridine triphosphate undergoes amination once again. I hope you remember that picture. When uracil undergoes amination, it forms a cytosine. So here the amino groups once again come from glutamine. This glutamine donates amino group and now it forms the CTP molecule. So here you will find that water molecule is also participating. The glutamate is formed. One oxygen goes to glutamate, and now the cytosine triphosphate or cytidine triphosphate is formed by showing amination, and the amination is done due to entry of glutamine. So glutamine donates that amino group and UTP gets converted into CTP. Now student, cytosine is a part of, or it's a pyrimidine present in DNA as well as RNA. So here now the role or the step which is very important is how ribose sugar gets converted to deoxyribose. But before that, have a look at this. This particular step is very, very important that we will be learning once again for your revision also. But remember, this uridine diphosphate is the common intermediate. Now what happens is this UDP undergoes the very major change, and that is deoxyuridine diphosphate. It means this UDP, which is attached to ribose 5-phosphate, is now going to get attached to deoxyribose sugar. So the enzyme here is ribonucleotide reductase. The name to remember is very easy. The enzyme is ribonucleotide reductase. Now, if you just visualize what is the structure of ribose and deoxyribose sugar. Ribose shows OH at the second carbon and deoxyribose sugar shows only hydrogen at second carbon atom position, and that is why we call it as a deoxyribose. So here the formation of deoxyribose sugar is very important, and that takes place due to the activity of ribonucleotide reductase. How? By the activity of the very important enzyme, and that is thioredoxin. This is the thioredoxin protein, you can say. This is the thioredoxin which shows the sulfhydryl bond. This thioredoxin shows or undergoes oxidation, and it undergoes oxidation and it forms the thioredoxin molecule. So hydrogen is now removed here. The hydrogen is removed and the reduction of ribose to deoxyribose takes place. This separately I will be showing you little later, but just remember, due to the involvement of thioredoxin and its oxidized as well as reduced form, it undergoes from its reduced to oxidized form, and it donates the hydrogen for forming or for the construction or for the synthesis or the formation of deoxyribose sugar. Now this UDP gets attached to that deoxyribose sugar, forming deoxyuridine diphosphate. But now this DUDP is very unstable because uracil is not a part of the DNA nucleotide. Hence, immediately it gets converted into DUMP. One phosphate is released, and now very important substance comes in the picture, and that is N5N10 methylene tetrahydrofolate. This is also again a specific protein molecule, and here this methylene THF, that is methylene tetrahydrofolate, gets converted into THF, and now it forms the thiamidine. Here the methyl group is now donated to the pyrimidine ring, and as we have seen that thiamine is nothing but the methylated uracil. So uracil is otherwise also having 2,4-dioxy molecule structure, and now it gets converted into the methylated one, forming the TMP, thiamidine monophosphate. This D indicates deoxyribose sugar. Remember, it is not double or divided or whatever, it is D for DNA or deoxyribose sugar. So like this, the pyrimidines are formed, and the pyrimidines are always, I hope you remember, they are unable to exist as a very separate independent nitrogen bases. They have to be in the form of minimum at least monophosphate structure, then only they are stable. So here you get to see the formation of the UMP, which we have seen here, then UDP. UDP gets converted into UTP. UTP gets converted into CTP. This CTP further may get converted if required to CDP or CMP as per the required. Here also, when it is going to be the part of DNA molecule, even the cytosine get attached to the deoxyribose sugar, which is formed through this way only. So UMP then gets converted into the CDP and CTP by getting associated with deoxyribose sugar. But why this particular part is highlighted? Just with one reason, that is the thiamidine or thiamine is the exclusively obtained in DNA, and for it is very essential to have methyl group in its structure along with deoxyribose sugar, and that is how the complete pathway takes place. So here I hope now that you must have understood the structures very well. When I will put the content on your classroom, you can see once again and study it, because the biosynthetic pathway is the major part of the entire chapter. Now, just for the reference here, I'm going to mention this salvage pathway. Now, this salvage pathway, when you look at this, see, salvage pathway is a secondary pathway, you can see, as far as the time and energy is required. Now, salvage pathway of any purine or pyrimidine synthesis is very quick, very short time, and less expensive. This is the second pathway which we had not discussed in detail last time. So here, the salvage pathway, what you're going to remember is, see, in salvage pathway, what particular feature? Pyrimidine base is ready-made available. It comes from various other reactions in which the metabolic process is completed. Say, for example, when the translation is done, RNA dissociates. Even when the DNA molecule, which is going to undergo the transcription and the replication also, there are excess of nitrogen bases present in the nucleoplasm. In cytosol also, after the metabolic pathways are over, free nitrogen bases are available. So these are recycled. Basically, the salvage pathway is nothing but the pathway to recycle the free nitrogen bases, which are the nucleotide component, and nucleotides can be formed fast, quick, promptly as per the requirement. But de novo synthesis has got its own metabolic importance because it shows the pure required constructive pathway, and it shows the production of many intermediates which are also essential. So in salvage pathway, this generally takes place in the cytosol. So pyrimidine base, which are available as a free pyrimidine basis, and they get attached to PRPP. Now this PRPP has to be activated. As we have seen in the de novo pathway, this PRPP has to be activated, ready to get combined with the pyrimidine base, and then the proper nucleotides are formed. And here the enzyme is pyrimidine phosphoribosyl transferase. So pyrimidine phosphoribosyl transferase is able to produce pyrimidine nucleotide. This pyrophosphate is free from the sugar, and at the first carbon position, pyrimidine base is attached directly to form the complete functional pyrimidine nucleotide. Now, salvage pathway, you may think that why then cell do not follow always the salvage pathway. See, student, it is like that. For the salvage pathway, ready-made pyrimidine base should be available. And these pyrimidine bases depends upon the other metabolic processes. The quantitative availability is not 100% every time, and hence the cell shows the salvage pathway as a secondary pathway, even though it is faster and less expensive. Now it is like that. See, suppose if I want to have few things, if the other spare parts are not available, then what do I do? The accessories are not available, then what do I do? You have to manufacture it. But if after utilization of manufactured products, if I have got excess of the spare parts, then I can use that for recycling. And hence, de novo pathway is the basic biosynthesis pathway which occurs in the liver, and in plants it occurs in plastids, mitochondria, as well as cytosol. But in animals, if you see, basic site or the primary site of nucleotide biosynthesis is nothing but the liver. From liver, it is transferred to blood cells, and from blood cells, it goes to tissues and cells, and then the cells incorporate that in the nucleoplasm where they are required in the highest amount. Now, just to give you a few points, that basically DNA replication and transcription needs lots of purines and pyrimidines. And DNA replications occurs only during the division, only during the cell growth, or you can say multiplication. Whereas transcription is a complete continuous process, very high rate and a continuous process where the purine and pyrimidine requirement is much higher. So to provide the required amount of purine and pyrimidine nucleotides, it is very essential to have de novo biosynthetic pathway properly going on and with a very proper regulation also. The salvage pathway is an process which fulfills the emergency need, and where the activated pyrimidine base gets attached to the respective sugar and it forms the required type of nucleotide. Okay, so this was about the first part. So I hope students you have understood that this was partly done last time. Now the next file I will come to. Now don't get surprised to see my handwritten content. This I have done because these are the things which I want you to understand in detail. Few things are images and few things I have written down. Now here, look at this. This I was telling you here. Okay. Now this is the one. Just check. Okay. This, if I tell you properly. 1 minute, let me test. Okay. Now, if you're looking at this screen, see. Now, students, remember, when you will be writing the answers or the assignment, I do expect these structural changes to be given, the simplest part, at least for certain simpler reactions. Not for biosynthetic pathway like de novo pathway, it's okay. But if you understand the structural changes, you will be remembering the pathway very well. Now here, see what is shown, ribonucleotide diphosphate. So you remember that UDP which I had mentioned. Now these all are comparatively, like you can say, the interconvertible purines and pyrimidines with each other. So here, this ribonucleotide diphosphate. So you take any typical ribonucleotide diphosphate. So here, these are the two phosphate groups, what we used to call it as a pyrophosphate in earlier one. And this is the nitrogen base attached to the first carbon position. Okay. Now here, when this ribose sugar has to get converted into deoxyribose sugar. So as you know, see, oxygen is missing here. So how it occurs? It occurs through a activity of an enzyme ribonucleotide reductase. What this enzyme does, it converts this ribose sugar into deoxyribose sugar via the activity of the important protein, and that is thioredoxin. So this is a coenzyme, or you just can say as a protein. So thioredoxin, which is in the reduced form, undergoes oxidized form. It means oxidized, it means what? The electrons are removed here, and oxygen from ribose sugar is taken out. So electrons are less now, and the number of protons will be more. So here it gets converted into the reduced form after the oxygen is removed. So the enzyme here is thioredoxin reductase. So conversion of reduced form into the oxidized form, once again oxidized form to reduced form. This cyclic process is under the regulation of thioredoxin reductase. Once the thioredoxin is in the oxidized state, how it gets proton once again to show the reduced form. So here the NADPH, which is the proton dependent process. So NADPH donates the hydrogen and it gets converted into NADP. This proton is now used by oxidized form to get converted into reduced form. Now students, once again here, listen very carefully. The reduced form and oxidized form are interconvertible, and they are interconvertible due to the activity of an enzyme, thioredoxin reductase. Now this thioredoxin reductase is a key enzyme for the formation of thioredoxin reduced structure and thioredoxin oxidized structure. The oxidized form obtains the proton or hydrogen from NADPH, and it gets reduced, it gets converted to reduced form and back in the cycle. But the key enzyme here is nothing but the ribonucleotide reductase, which converts this ribose sugar into deoxyribose sugar. Once this deoxyribose sugar is formed, the base remain attached to it, and what is formed is DNA nucleotides, that is DADP, DGDP, DCDP, and that is TDP, or you can say thiamine is otherwise also DNA nucleotide, so D is not mentioned, but that's fine. Okay, so this is the way how the sugars are converted to the other format. Now I had said that I will be showing you the slide where this nucleotide, how it gets converted into the thiamidine type. It's the same sort of, now you can understand here, UDP shows conversion into this DUDP. This is the DUDP. And again, as I had shown earlier, it is ribonucleotide reductase with the incorporation of thioredoxin to thioredoxin oxidized form. So this is the reduced form and this is oxidized form. Now deoxyribose sugar is available. So uridine is attached to deoxyuridine diphosphate. So formation of deoxyuridine diphosphate. Now this UDP gets converted into DUMP, that is deoxyribose uridine monophosphate, and now this DUMP gets converted into thiamidine, or this thiamidine monophosphate by entry or by participation of a very important compound, and that is N5, N10. This is the nitrogen position they are telling, and methylene tetrahydrofolate. This tetrahydrofolate donates the methyl molecule, and now the enzyme is thiamidylate synthetase, and now finally thiamidine monophosphate is formed, and as per the requirement it is incorporated in the DNA molecule during its replication only. During its replication, thiamine is required only for the replication of the DNA molecule. And I hope you understand, when transcription takes place, instead of thiamine, there is a uracil substituted. So now here the conversion of, as I said that I had written it for you in my own handwriting. I believe that once you write, you remember better. And here I'm showing you the conversion of ribonucleotide to deoxyribose nucleotide with the activity of the required enzyme. So this was the same. Now here the same, but this I want students you all to read. Now here some information for you all. That synthesis of deoxyribose or deoxyribonucleotides. Deoxyribonucleotides, the building blocks of DNA, are not synthesized by de novo synthesis. Deoxyribonucleotides, the building blocks of DNA, are not synthesized by de novo synthesis, but are formed from their corresponding ribonucleotides. So nitrogen base are synthesized by biosynthesis, by de novo biosynthesis. But when it comes to the DNA nucleotide, it has to be synthesized by formation of the deoxyribose sugar to which the bases are attached. So ribonucleotides undergo the ribonucleotides undergo reduction at C2 position. Reduction at C2 position, which I had shown you that they undergo at C2 position, where the hydroxyl group on ribose moiety is replaced by a hydrogen atom. The enzyme ribonucleotide reductase catalyzes the reduction reaction for all 4 ribonucleotides of DNA molecule. And when you look at the reaction mechanism, you will find that ribonucleotide reductase complex is a multi-subunit enzyme possessing allosteric site. This word is very important. Allosteric sites you will be learning in the enzyme chapter, but allosteric site enzymes, I mean the enzymes which are having allosteric sites, they have different sites for the substrate as well as the activators or inhibitors. The sulfhydryl groups, that is SH, of the enzyme provide the hydrogen atoms required for the reduction. Thioredoxin oxidized in the process is reduced back by NADPH dependent enzyme, that is thioredoxin reductase, and is recycled again. The activity of ribonucleotide reductase is regulated by allosteric modulators, which ensure that the synthesis of deoxyribonucleotides occur only when cells are actively synthesizing DNA preparatory to cell division, that is DNA replication process. So that ribonucleotide transferase, or that enzyme which converts, or ribonucleotide reductase, that converts ribose sugar to deoxyribose sugar, is required to be active only during the DNA replication process, especially when thymidine nucleotide has to be formed. Okay. So now next is the one which I had earlier also showed you. Now I want you all to have a look at this particular picture. The content which I've explained you is already there. I'm going to send this PDF to you in a group, but just have a look at this here. This is the uracil, which is the major or basic nitrogen base. It gets attached to the first carbon of ribose sugar and it forms ribose nucleotide, that is uridine. Now this uracil shows the amination and it forms cytosine, which is 2-oxy-4-aminopyrimidine, and when it undergoes methylation, it forms the thiamine, which is 2,4-dioxymethylpyrimidine. So here amination is by glutamine and methylation is by methylated tetrahydrofolate. So here you can understand how they are converted from uracil or uridine monophosphate or diphosphate further. Okay, so the same one for you all in my own written format, that UMP to UDP by kinase, UDP to DUDP. Here it is by the ribonucleoside reductase and thioredoxin from oxidized to reduced form conversion. Here the ribose sugar gets converted into deoxyribose sugar. Uracil is attached to the deoxyribose sugar, so deoxyuridine diphosphate, and it releases one phosphate, inorganic phosphate, to get converted into DUMP, and DUMP now shows the activity of methylated tetrahydrofolate N5N10 methylene tetrahydrofolate, which donates this methyl group, and ultimately it forms the deoxyribose nucleotide with one phosphate, and that is thiamidine monophosphate, which is deoxythiamidine monophosphate, and exclusively DNA nucleotide pyrimidine. So there are n number of things, students, to remember and to understand what exactly happens in the de novo pathway as well as in the salvage pathway. There are many key steps which shows the you can say qualitative significance, and in that reference, let us study a very important part of this chapter, which also I have made a note for you all, and that is nothing but the regulation of pyrimidine biosynthesis.
Ma'am.
Hello.
Sorry to interrupt, but you told me that last 5 to 10 minutes you want for discussion.
Acha, so only 5-10 minutes are left.
10 minutes are left.
Okay.
And, suppose you want to discuss something, then
So I'll just complete this, just a mention regulation, and then I will ask for the discussion. Okay.
Yeah.
Because this is they have already aware, I hope.
okay.
Okay. So now let us see the regulation of pyrimidine biosynthesis. Now, students, very important thing which I want you to understand is what is regulation. So regulation of any metabolic pathway is an important concept which the physiology student must understand. Now, when you consider any metabolic pathway, it shows either anabolic or catabolic. Now, this pathway is operative mainly to form the required product. But there has to be some control. There has to be some control over every metabolic pathway. Now, students, if I give you your own example, see, there are n number of factors which activate you and deactivate you. Now, suppose if you are thinking about coming to college, then there are many factors which will motivate to come to college and study. There are few factors which will inhibit you to come to college and studies. So these factors are the regulatory factors. So when you look at the cell metabolism, there are always the regulatory factor exist which are very essential to maintain equilibrium. So to maintain the cellular equilibrium with respect to every metabolic product or intermediates, there has to be some regulation. Now this regulation is basically on the activity of enzymes. The catalysis process shown or the role played by the enzyme is very important, and these enzymes has to be kept under the regulation. There are n number of factors which regulate even this enzyme activity, but that we are not going to discuss right now. You have just heard like pH, temperature, substrate concentration, and so on. So these are the actual regulatory factors for enzyme regulation. But how these enzyme regulate the biosynthetic pathway, one has to know this. So the quantitative regulation of biosynthetic pathway is essential for proper functioning of nucleotide mediated metabolic processes. Now say for example, DNA replication is going on. So that has to be regulated by the enzyme activity, and then only it will be in equilibrium. So regulation is must, and it's a key point to have the equilibrium at the cellular level. So the first important part you have to remember is the quantitative regulation of biosynthetic pathway is essential for proper functioning of any, say, nucleotide mediated metabolic process. Regulation occurs via effectivity of functional unit. What we call it as the functional unit, nothing but the protein, domain protein of certain enzymes or certain proteins. So what is first regulating steps in pyrimidine de novo biosynthesis? The very first step, which I had asked you to focus very well, the enzyme carbamoyl phosphate synthetase 2, CPS2. This is the first step, and students, you can see here, the step which I have shown in red. The step which I have shown in red here, you can make out that the carbon here, the carbonic acid, glutamine, it gets phosphorylated and it forms the carbamyl phosphate. This is the first step. This enzyme is always a key enzyme, and this is the activity of CPS2 is regulated by formation of this UTP and UDP. It is inhibited by UTP. If excess of UTP are formed, the activity of this enzyme is inhibited. If excess of UDP is formed, the activity of CPS2 is stopped. It slowed down, and hence the further cycle is also slowed down. But the same enzyme is activated by ATP molecule and PRPP. If more PRPP are readily available, then the cycle rate increases. So activators are ATP and PRPP, whereas inhibitors are UTP and UDP. Then the next, which is the very, very important step. Now here the second step, and the enzyme here is aspartate transcarbamylase, ATCase. Here, this is also showing the regulatory steps. This enzyme catalyzes step 2 of de novo synthesis. It is inhibited by the end product CTP, the last one, that is CTP. When it is formed from UTP or UDP, it is called as a feedback inhibition. So if excess CTP are formed, then the rate of activity of ATPase is slowed down, and that is how the carbamyl aspartate, this aspartate contribute major portion, and this step is slowed down, and hence the ring is not formed with a higher rate. So the step is slowed down. So aspartate transcarbamylase is under the regulation of feedback inhibition by CTP. Okay, so this is the second key enzyme. And the third one is the step 6, which we had seen, and that is orotidine monophosphate decarboxylase, where the carbon dioxide is removed. Orotate monophosphate is, or orotidine monophosphate is converted to uridine monophosphate, and the enzyme is OMP decarboxylase. This enzyme is also inhibited by UMP and CMP, which are intermediates. So here, if excess of UMP are formed, decarboxylation do not occur, so OMP cannot get converted to UMP and the regulation takes place. So 3 steps you have to remember for de novo synthesis, and these all 3 steps act as a regulatory steps, and that is how overall regulation of pyrimidine biosynthesis takes place. And this is a very, very essential mechanism. Generally what we do, we learn biosynthetic pathway, we learn metabolic pathway, but we are not much aware about how it is under the regulation of various components. So students, here I've stopped my content delivery, and you can ask the questions. You can ask the questions, you can talk, you can unmute yourself, and you can ask me the question. Okay. So students, you are welcome.
Yeah, if you have any questions, students, you can unmute or you can put it in chat box.
Chat box भी open कर.
Ma'am.
हाँ ठीक. हाँ. Okay.
Ma'am, this salvage pathway, this will only be operated when there is enough pyrimidine base which are formed by de novo pathway.
Correct. De novo pathway nahi. See, when there are enough pyrimidine free bases are available as an end product of various metabolic processes. Now, say for example, when there are secondary messengers and all other components are participating in many reactions, once the reaction are completed, they show dissociation from the substrate. And
yes,
if this dissociation results into enough number of free nitrogen bases, as I said, free nitrogen bases are very unstable. They have to get attached to the sugars to get them stabilized. And immediately the salvage pathway goes on. Salvage pathway is a secondary as far as the requirement fulfillment is concerned. So this salvage pathway is necessary. Otherwise, how the recycling will take place? Because if the nucleotides show degradation, now say for example purines, when purines undergo total breakdown, they form uric acid. And uric acid, if it is accumulated in the body, it results into the disorder. So
Yes.
to prevent the excess storage, the salvage pathway has to be operational.
Thank you, ma'am.
Anybody else? First of all, student, do you find it digestible? These nucleotides, do you find it digestible? You must, as I'd already said, it's going to be the pure chemistry. But believe me, once you do it with very, very proper observation of the structures of nucleotides, then how the step by step the structural changes takes place, then you will be able to write it properly. Don't worry. Okay, anybody else?
I think Abhijit had got disconnected.
Huh.
Are you there now?
Anybody is there? See, doubts are possible only in two conditions you know very well, na? If you have understood everything, or if you have not understood anything. So students, I hope positively I take that you have understood everything. Now next lecture when I'll start with purine biosynthesis. Purine biosynthesis, I will be going very slow because purine de novo biosynthesis is again a lengthy process, lengthy metabolic pathway to understand, and lots of chemicals, name, lots and lots of components involved. So it will take one complete lecture only to explain purine biosynthesis. So students, this PDF I'll be putting on the Google Classroom, and please study that. If possible, you can take the printout and do the practice. Anybody still here? Girls, are you there or sleeping?
Abhijit had some electricity issues.
हाँ ठीक है, but other anybody, nobody's ready to say anything.
Sugyani is now there.
Okay. So I shall take your leave, and if you have a doubt, you can even put on a WhatsApp group, no problem. You can put your questions on WhatsApp group, and then I will give the answer, definitely.
Ma'am, can you stop sharing that screen?
Okay, I'll stop sharing screen.
So that
Okay.
ask Ashwara to join it.
Yeah, okay. So
Ma'am,
let us, yeah. Okay.
Yeah, bye ma'am, take care.
Bye, take care. And students, I will meet you again day after tomorrow, right?
Yes.
Yes, thank you so much.
So I'm switching off the recording.